When a bacterial infection is established in the body, the purpose of the immune system is to control or eradicate it. The initial reaction of the immune system to an infection varies depending on the site which has been invaded or the nature of the invader. The overall effect of the initial immune reaction is inducing phagocytes to migrate to the site of invasion. Once they reach that site, the phagocytes are activated and begin digesting and destroying invading bacteria. Once activated, they also produce cytokines, which further activate other cells of the immune system. Despite studies showing increased inflammation and depression, little is known about the mechanisms through which inflammation may trigger depression, or whether inflammation is simply a consequence of the experience of depression. Cytokines involved in the inflammatory response have been linked to stress-related disorders such as depression and anxiety, specifically the release of cytokine interleukin-6. IL-6 has many functions within the immune system depending on which type of tissue it is acting upon, thereby contributing to its role as both an inflammatory and anti-inflammatory cytokine. Within the peripheral and central nervous systems, IL-6 can act as a neuronal growth factor, leading to neurite development and nerve regeneration. However, this is highly dependent upon circulating IL-6 levels as well as the inflammatory state of the animal. Because IL-6 acts on so many different target tissues throughout the body, dysregulation can precipitate a multitude of events relevant to stress disorders. While it is unlikely that IL-6 is acting alone to trigger the symptoms of depression, blocking the effects of IL-6 can prevent further escalation of inflammatory responses. This paper explores the theory that depression and anxiety are linked to an increased release of peripheral cytokines and that this increased release may be a pre-existing genetic factor. The experiments conducted in this study demonstrate that emotional responses to stress can be generated or blocked in the periphery and potentially offer new methods of treatment for stress disorders. To explore the source of this potential genetic factor, the researchers determined that patients with major depressive disorder consistently showed higher levels of certain inflammatory cytokines, specifically cytokine interleukin-6. Many cytokines are regulated by stress, but the most upregulated cytokine in mice that developed a susceptible depressive behavior pattern after chronic stress is IL-6. To address whether systemic inflammation contributes to stress vulnerability, the researchers used several types of stressors: repeated social defeat stress, witness defeat, and chronic variable stress, where social defeat is defined as losing a confrontation among conspecific animals. In susceptible mice, chronic social subordination will lead to depressive behavior, and resilient mice will not develop this kind of behavior. A relationship between these peripherally derived cytokines and stress-related disorders has not yet been confirmed, since it is unknown if inflammatory status predates the onset of disease or whether it contributes to depressive symptoms. The researchers' hypothesis was that pre-existing differences in the sensitivity of an individual's peripheral immune system dictate their subsequent vulnerability or resilience to social stress. The materials used were all male mice, plexiglass hamster cages with dividers, volunteering human participants, either healthy or diagnosed with major depressive disorder. Many types of software was used to measure cytokines released, and the severity of depression was quantified by the QIDS scale. Repeated social defeat stress. Mice were exposed to an aggressor mouse for 10 minutes daily over 10 days and were then relocated to a new cage each day and defeated by an aggressor to prevent stress habituation. Witness defeat was defined as mice witnessing the other experimental mice experiencing the segments of repeated social defeat stress but did not have physical contact with the aggressor mice. Chronic variable stress. Mice were exposed to 21 days of variable stress, rotating between foot shocks, tail suspension, and restraint stress. The stressors were applied in the above order for a total of 21 days. IL-6 mice were given daily injections of mouse anti-IL-6 or a saline vehicle as a control. The differences were compared using a t-test, and multiple groups were compared using an ANOVA with a statistical significance set at p is less than 0.05.
This study found that leukocyte-derived IL-6 is predictive of susceptibility to stress. In the experiment, an aggressor was applied and exposed to the subjects via a repeated social defeat stress, and after 20 minutes, the cytokine and chemokine profiles were examined and was found that IL-6 was the only cytokine significantly elevated in animals that would later develop a susceptible phenotype. High levels of IL-6 were found to be negatively correlated with social interactions because of the RSDS. In susceptible mice, along with high levels of IL-6, were also found high levels of IL-1 beta, but not in resilient mice, and this was not found to be statistically significant to the study. Chemokines ligand 1 and 2 were similarly elevated in both groups of mice and was found to also contribute to whether the mouse is stressed. However, this was not a contributing factor. IL-9 correlated with social avoidance behavior following stress, but is not regulated by stress. In a second group, an in vivo course of plasma IL-6 levels before and throughout exposure to stress were examined and found that susceptible animals had higher levels of IL-6 within 20 minutes after their first contact with an aggressor and subsequent defeat response, and the levels remained in their system up to 48 hours after. Mice were determined to be susceptible or resilient based on immune differences in stress-naive mice in accordance to their responses after RSDS exposure from an ex vivo leukocyte immune challenge, where basically leukocytes were isolated and counted 4 days before physical interaction from the RSDS aggressor. Higher levels of circulation leukocytes were exhibited by the susceptible mice. The results were validated as clinically relevant by bringing in human subjects. IL-6 levels and the severity of depression for patients who had treatment-resistant MDD were measured and quantified. The results found were that patients with MDD had higher serum IL-6 than a healthy control. The experiment took a second group of patients with treatment-resistant MDD, and the effects of antidepressant treatment on circulation levels of IL-6 were examined, and the severity of their depression quantified as well. This group led the experiment to find that standard antidepressant treatment did not lower circulation levels of IL-6. A different group of subjects received 10 days of RSDS followed by 35 days of a treatment called imipramine, but it was found that imipramine did not lower the circulating levels for IL-6. A different set of mice was exposed to 21 days of chronic variable stress to see whether effects of stress and IL-6 levels generalized to other stress patterns. It was found that levels of IL-6 were increased 24 hours after last stress exposure due to the CVS. In a second group, an emotion stress was examined as a possible trigger to the increased levels of IL-6 by exposing and undergoing RSDS to the mice without physical contact with the aggressor, and it was observed that IL-6 levels elevated in the mice compared with controls 30 days after the last stressor, when the phenotype of social avoidance typically emerge. It is important to note that in this study, individual differences in susceptibility to RSDS were not due to physical injury because there was not found to be any differences in the number of wounds and nor correlation between number of wounds and social interactions, and that general sickness behavior was ruled out to being altering the social interactions by taking core body temperatures 10 days after RSDS. To further rule out whether general hypothalamic pituitary adrenal stress responses were occurring from the high levels of IL-6, the susceptible and resilient groups received similar levels of trauma-related stress indicated by the similar increase of corticosterone levels. So it has been found that there are individual differences in the sensitivity of the PNS in response to social stress, and there are pre-existing factors that can tell whether there is a greater risk for developing a stress-related disorder. Being more prone to developing a stress-susceptible phenotype showed higher levels of circulation leukocytes, due to the monocytes that in turn produce the IL-6. This experiment has noted that it is the first study to show that there is a predictor in individuals in regards to vulnerability to a social stressor prior to its exposure and stimulation of that stressor that leads to depression-like phenotypes in vulnerable individuals. Elevated serum IL-6 and circulation leukocytes in stress disorders have been found to generally follow depression and anxiety diagnoses in humans. A growing number of humans with depression and anxiety are resistant to many antidepressant treatment. This could lead to further research and treatment for depression in humans in regards to controlling monocyte production and specifically interleukin-6.
The first problem were several different types of stressors were used to test a systematic inflammation effect on stress vulnerability. My critique would be the results in different levels of inflammatory cytokines include IL-6. What should be done differently was the inflammatory status response measurement for each mouse that was tested should be grouped by the type of stressor used because different types of stressors could result in the different cytokine levels for each mouse. Second problem would be that the two different tests that were used to measure the depressive symptoms in mice. While the QIDS would be a good predictor for probability of inflammatory cytokine levels, IL-6 was also found to be a more accurate predictor of stress vulnerability. Also, using two different types of depression tests could result in different measurements of depressive symptoms. What should be done differently is if only one test for determining the severity of depression was used for both before and after the antidepressant treatment, a correlation could have been determined by evaluating cytokine levels. The third problem is the results of the experiment were inconclusive. The inflammatory response did not determine whether it was a precursor or of depression or just contributes to depressive symptoms. The different tests used throughout the experiment has led to the misapplication of these experiment results. And what could be done differently would be that better organization could have resulted in being able to predict whether inflammatory cytokines are precursors of depression or just responses.